Yes, I was asked to talk about tuna migration, and I guess I have two messages today to give. One is that there's many kinds of tuna, and the other is that they act differently. They're not all the same, and also the same species of tuna can act differently depending on what part of the ocean it's in. And that's an interesting concept to a lot of people. They just think, well, these ones do that, and they're all highly migratory. But I'm going to dispel that a little bit, but also reinforce it in some ways. Okay, so here we are. They've got the Pacific Ocean. This is the realm in which to do my research, the whole thing. And theoretically, there's no barriers to movement within this whole region for tuna as long as they stay within the warm water. Nevertheless, there is some separation. Okay, I like to start the talk with the tuna and billfish family tree. These are all members of the same suborder: marlins, swordfish, tuna, mackerel, onos. They are all related. And today I'm just going to be talking about what I call the true tuna, the tuna of the genus Stunus, which there's 9 species worldwide. You'll see some familiar faces up there, Aku and Onos, things like that. But now we're just going to talk about the tuna, of which there's 5 main types that are important commercially in the world. Actually, 7 species, because bluefin tuna, there's actually 3 species of bluefin tuna. A lot of people don't know that. But the Atlantic species separate from the North Pacific species. And then there's something called a southern bluefin tuna you find below Australia and New Zealand. They're similar, very similar, but very distinct species. And then you have albacore, big eye, yellowfin, and skipjack, or the aku. And these are the ones that make up the bulk of all the commercial tuna sales in the world and what you get in restaurants and markets. But if you look at these species, there's 2 main kinds that we categorize as in science. Those are the temperate water tunas, the bluefins and the albacore, and the tropical tunas. And I'll just let the cat out of the bag. And the main part of my first lesson is that the temperate water tunas are highly migratory animals. They do migrate. And by migrate, I mean they have specific movements that they make at certain points in their life cycle, or seasonal movements, just like ducks migrate south during the winter. That's what I call a migration. There's a certain purpose for it, either to spawn or to feed, and they do it regularly throughout their life cycle, repeatedly. Now, the tropical tunas are very different. The big eye, yellowfin, and skipjack, and particularly the yellowfin and the skipjack, they're not highly migratory per se. They can live out a large part of their life within a fairly small area, we find. So that highly migratory sort of name doesn't really apply. And big eye tuna, which make up the bulk of sashimi sales worldwide, very important tuna, actually, they're kind of a combination of the 2. So talk about albacore. This is a North Pacific albacore. There's a South Pacific albacore stock and a North Pacific albacore stock. They definitely migrate. Doesn't show it here, but the 1-year-olds, which isn't even shown, they'll migrate from Japan over to the west coast of the U.S. and then spend some time growing and feeding, sometimes moving out to the Central Pacific and then returning anywhere between British Columbia and Baja, Mexico. And if you're from the West Coast, you know albacore is a big deal there. Beautiful. Sport fishery, commercial fishery, Canadian fishery. They mature maybe 5-6 years of age, and at that stage, if they're on the West Coast, they have to migrate back over here to the Central and Western Pacific to spawn. That's the only place they do spawn is here in Hawaii and also south of Japan. So that's And the albacore in the Atlantic act the same. They have migration routes to spawning areas and feeding areas. These slides, by the way, are courtesy of John Childers at the NOAA facility in La Jolla, National Marine Fisheries Service. Customers. He's done a lot of tagging of albacore on the West Coast, and these are movements of albacore tuna from archival tags. And archival tags give positions of the animal, or you can derive positions of the animal from those tags. And you can see these movements out into the Central Pacific, and then even trans-Pacific migrations and movements of albacore from the West Coast back to Japan. Now, bluefin tunas are another temperate water tuna that definitely migrate. This is a very old representation of the Western Atlantic and the Eastern Atlantic bluefin stocks. And this is not really true. They don't really cleanly divide in the middle of the ocean for management purposes. That was someone's dream, and that obviously doesn't happen. That's what managers would like to happen. But the truth is more similar to this picture, where you can see that the Western Atlantic population of bluefin tuna overlaps broadly with the Eastern Atlantic population of bluefin tuna. And in truth, both stocks occupy the whole Atlantic Ocean. Which really, the Atlantic is kind of small compared to the Pacific. And bluefin tuna have kind of evolved, well, they have evolved to take advantage of both sides of the ocean that they live on. It's like they're too big for one side. Yeah, kind of interesting phenomenon. But see, so this eastern population spawns throughout the Mediterranean Sea. The western population spawns only in the Gulf of Mexico, but now they're finding some spawning out in this region also. But fish from this population will actually visit here and back and forth. They find the tagging, but they are distinct stocks. Okay, here's some more on bluefin. As a true migrator, you can see this is a classic example of shows the spawning grounds of the North Pacific bluefin tuna. They migrate to the West Coast and often spend years, 4 or 5 years, feeding and growing in the same areas as the albacore, eventually returning to spawn. So that's a true migrating species of tuna. I mentioned that bluefin tuna use both sides of the ocean. It's been shown that there's a reason for this, and there's a large feeding area here off the east side of Japan, and apparently when the stocks of Japanese sardine are low, those fish will move to the west coast here and take advantage of the anchovy and sardine stocks of America, back and forth. So like I said, they're an animal that's almost too big for one side of the ocean, and they'll take advantage of both to grow and feed. Here's another example of bluefin tuna migration you probably saw. When the Fukushima nuclear disaster occurred, fish were in that area were picked up that signal of that strontium, and bluefin being migrators across here, that signal was detectable in their bones. And I'll just say that it's not particularly harmful in a health standpoint because it's such a small amount, but it is interesting that it's detectable and has been used in other studies to age fish successfully from way back when they were doing above ground nuclear testing in the 50s and 60s. Some of those fish that were affected by that are still alive. Again, it's not a health problem. It's an interesting tool that scientists are now using to age long-lived species. Okay, so that's the migrators, the albacore and the bluefin tuna. Now I'm going to get into the tropical tuna, skipjack and yellowfin tuna, and talk about their movement characteristics. These boats are here because these are boats that were used during large-scale tagging projects. This is where tunas are caught and the plastic dart tag is put into their back with a number on it, and they're released to see how fisheries react in catching them. So when fishermen catch them, they report the tag, and you get a point of release and a point of recapture. And the point of this slide is that during large-scale tagging projects in the South Pacific, mostly on skipjack, but also with some yellowfin, like 40,000 yellowfin tuna, most of the movements recorded were just pretty much east-west. We didn't get a lot of those fish. Actually, only one that was recovered in Hawaii. And that alone is kind of interesting. The analysis of that tagging data led to some interesting papers. I'll mention this one, Kleiber and Hampton. I looked at the movements of tagged tuna and found that in the Solomon Islands, which is a double chain of islands, also surrounded by a line of fads that were used to support Poland line and purseine fisheries there. They found that the fish didn't move out of that area as quickly as they would normally, suggesting that there's an influence of islands and also man-made things like fish aggregating devices, which are buoys put in the water to help fishermen because the tunas go around these buoys. So that both man-made structures and islands themselves can retain fish or affect their behavior to some degree. And likewise, this paper, Seibert and Hampton, 2002, analyzed a huge amount of tagging data within this area where the main fishery exists, and found that yellowfin and skipjack don't really migrate or move large distances in this area. It's more like, on average, a matter of hundreds of miles, not thousands. So that their lifetime may be spent in 2 or 3 countries' zones and not widespread. And the reason this is important is it suggests that management of tunas may not, it needs to be international between countries, but that management can benefit perhaps of some sub-regional effort. And look, if you know enough about your fish. Okay. So it begs the question of interaction and movement between areas. And this is a question that countries on the edges of the periphery of this huge area of tuna, where more than 50% of the tuna in the world comes from commercially, want to know, like Japan, east coast of Australia, small countries, Niue, French Polynesia, and Hawaii. Like what are large-scale removals of those fish by large-scale fisheries doing to these outer fringe areas? Is there an influence or not? This is something we call connectivity. How are the different regions of the Pacific connected biologically? Let's say. Okay, now I'll move to tagging projects we've done inside Hawaii. This is a project I was involved in called the Hawaii Tuna Tagging Project. A nice experiment where we were able to tag 16,000, 17,000 tunas, big eye and yellowfin, roughly half and half of each in the Hawaii zone, and then let them go off and get recaptured and see what happened. This was a PowerPoint, but now it's just a PDF, so I have everything parallel on top of each other. But basically, tagged yellowfin from Midway did show some wide dispersal and recaptures in Okinawa, Japan, and for seamounts, and also back into the Hawaiian Islands. We had a single recapture from all these thousands of yellowfin that was made in the Eastern Pacific, Baja, Mexico. And but the main story was that I feel was that within the main Hawaiian Island group, where we tagged thousands of tuna, yellowfin. Here you can see a fish with the tag in it. They were virtually all caught within that same Hawaiian Islands area. And this surprised people, but it is what it is, and got us thinking, what's going on here? And I'll just mention that it was compared to big eye tuna, it was very different. You can see those blue lines. This is the big eye tuna releases in the Hawaiian zone, and then subsequent recaptures in the zone and also out as they grew and were caught by longline fisheries and other fisheries outside of Hawaii. We've done other kinds of tagging in Hawaii. This is an example and kind of way of showing acoustic tagging. This is where you have these are fads, by the way, anchored buoys that the state puts out to help fishermen. Tunas aggregate around them, lie. Fishermen go and fish on it. It's a standard fishery enhancement method used throughout the world. So this is a way of helping smallboat fishermen just by anchoring these buoys, and tuna show up. We use these buoys as almost scientific research stations by equipping them with acoustic receivers. 60 feet below the buoy, and mounted these receivers on the fad, and then we tagged the animals with these pingers. They're actually transmitters. So once you put it in the animal, sew them up, they heal up, and then they go around transmitting this signal that's unique, so that they say that this is yellowfin 1057, and we know that fish was tagged on this buoy on such a day, and then whenever it gets close to these other receivers, it'll check in and be recorded. So this is an interesting way to look at fad use and visitation by tunas. And just an example, there were some fish that spent quite a lot of time around fads. There's others that just left, visited maybe one and then left the array completely, not to be heard from again. But as an example, here's the one that was tagged and recaptured, tagged on this fad, recaptured on this fad 4 months later. And during that 4-month period, it spent 25% of its life on a fad around Oahu, which is considered quite interesting. Certainly not the norm, but it does show that these structures can influence the behavior and the habitat of these animals quite a lot. You can read this through. It says tagged on CO fad, and then it just took off for a couple months, never heard from it. Then it came back into the Oahu fad array. See, we have fads on the other islands too, but they don't have receivers on them, so we didn't know where they went. Then it just came back in, spent a month between J and X fad, and then went by LL fad very quickly, spent a month around between MM and U fad on the Kaneohe side, where it was eventually captured again. So this is a look at a very specific look at fad residents, movement and behavior, but obviously you don't know what happens when they're out of the array. So to get at that, there's these things called archival tags, which I mentioned before. There's the stalker one coming out of there. It's also a surgical implantation. And what we did was we put sonic tags and archival tags in the same fish. So that if the sonic tag was transmitting and it was near a buoy with a transmitter receiver, we knew exactly where it was. And when it left those buoys, and if we got the tag back and downloaded the data, we're able to see where it went when it was away from those transmitter receiver equipped fads. So this is sort of behavior on the fad, behavior in open water, deep diving in the day, shallow night. And what we saw from these was that of the recaptures we got and the downloaded data, the fish was tagged here and then recaptured on the yellow arrow, and it wandered around Oahu quite a while, visiting fads or not, made excursion away from the island, but eventually came back. Here's another good example on the right. Fish tagged on the yellow arrow and recaptured in the red arrow. Again, wandering around the island, making some excursions away. then offshore, eventually coming back into the Big Island, where it actually made the mistake of going to a fad and getting caught. So, but the point of all this is that we have not documented broad scale movements away from the Hawaiian Islands for the yellowfin that we at least monitored. Going back to the Western Pacific equatorial fishery, there's been a huge amount of tagging down there. And this is something that I was involved in earlier in my career when I worked in the South Pacific. The South Pacific, now called the Secretariat of the Pacific Community, undergoes these large scale tagging projects, talking about 100, 150, 200,000 fish per experiment to seed tags out in the population of tuna and see how the fishery is impacting them, how the fish are moving, etc. So this is an example of the latest project that's tagged and released over 400,000 tuna, skipjack, yellowfin, and big eye. And you can see the colors: yellowfin represented in yellow line movements, green is skipjack, red is big eye. And again, mirroring the earlier experiments where you see a lot of east-west movement within 10 degrees of the equator, but not a lot of north-south, at least recaptures. Hawaii is up here somewhere. We don't see that connection so much there. So this suggested that maybe we should try a different technique of looking at movement of tunas. And that's when I hooked up with Dr. Jay Rooker and Dave Wells, and this project looking at Nursery Origin of yellowfin and big eye in the Hawaiian Islands. By Nursery Origin, that's the way that they talk about where that fish was actually born. Where's it from? So the way this is done is through examining the tiny ear bones inside tuna. Every fish has two of these sagittal otoliths, they're called, tiny little bones that grow at a regular rate. And in tuna, yellowfin tuna, we know that they deposit one ring of growth per day, every day of the animal's life. So it's an amazing way to age tunas. But they also will incorporate the chemical structure of the water they're in and the stable isotope signature of the water that they're in. And that will be sealed every day by another layer so that it's not disturbed by subsequent growth or movement to different areas. So it's very cool technology in that you can look at microconstituents of chemicals in the water or the stable isotope signature. The fish has two, so each fish was examined for both of those parameters. One otolith for chemical constituents and one for stable isotope signature. And the methodology is you have to sample a lot of different areas, and you mill down to the very center of the core of the otolith, representing the beginning of where that fish was when that fish was born, basically the early months of life. And it's not perfect, I mean, because obviously little fish can move a ways in a month, but if you do it for enough different areas and get unique signatures, then you might be able to talk about different origins of fish as far as location. And we're really happy to find that apparently Hawaii has a signature that was unique from all the other areas that we looked at. The Line Islands, Marshall Islands, Solomons, Philippines, etc. Again, it's certainly not perfect. I mean, in a perfect world, you'd sample lots of places, all over the place. But this, over 3 years of time, I took that slide out of the presentation, but over 3 years of time, we found this signal to be consistent, that there was apparently a signal for fish that had been started life in the Hawaiian or central North Pacific region of Hawaii. What was even more interesting is if we looked at fish, those are fish that were very, very small in sample. We tried to get them the next year, if they theoretically would have grown another year or two at this size that's very important to our local fisheries for sport and commercial and subsistence. This is size that people often like to go out fishing and catch, take home to eat. Virtually all of those, when the center core of their others were examined, also had that Hawaii signature. This suggests that local spawning is really important to our local fisheries. Now, the Big Eye story is very, very different. And we're just, frankly, now getting the manuscript that's pulling both the Big Eye and the Yellowfin. The Yellowfin work is published. The Big Eye Yellowfin story is in final preps for publication. And this is very different, where we don't sort of have that overriding local signature. And this is and a lot of it, the signatures we got overlap with the Line Islands south of Hawaii and some other signal that we didn't sample that I'll explain later. But this makes actually sense when you look at the life history of the animals, because yellowfin, we know for a fact that they spawn in Hawaii. They spawn right off Waianae and Big Island, Hilo. It's the summer run of big ahi we get. That's these animals here that every June, July, August, they're inshore spawning. And that's when they're also vulnerable to trolling. That's when we control them. And you see the big egg case there like that. This is a study I did years ago too on yellowfin spawning. It's 3 years of data, and actively spawning, reproductively mature females are found between May and September every year. And then the rest of the year they're mature, but they're not spawning. But for Big Eye, you catch right here in the Hawaiian Islands, they don't spawn typically, suggesting they have to go somewhere else to do that. Okay, these are the sampling areas from the new paper, and I'll just finish up by saying that we know that the temperate water tunas migrate to fulfill life history functions like spawning, feeding. Yellowfin and Big Eye have much more restricted movements. And in the case, people have taken some of this work to say that yellowfin are just resident here, resident there. I don't really like that word. They're not resident here. They just have a more restricted movement in their life history, because they can go to Mexico. We know that they can go to Japan, but on average they tend not to. So people talk about a local population. Well, it's sort of a you can mince words, but it's as a scientist I could say a localized population. And you find the same thing of this Baja Mexico from tagging work there and other parts of the Pacific. But it's not surprising, because the Hawaiian Islands are the most isolated island archipelago, major archipelago in the world. That's why we have so many endemic or local species of reef fish here. So many local species of birds and spiders and everything. They're so isolated. And that's true for even the ocean. There's not a natural current that sweeps from the equator up to Hawaii. Otherwise, we'd have a lot more of the reef fish species probably that they have that don't occur here. This is just a picture from a study showing Big Eye spawning areas. And you can see there's an area quite active spawning just south of Hawaii. And this is the Line Islands region. So based on that stable isotope analysis work that the yellowfin and the Big Eye, there's some support that our population of Big Eye that are around the Hawaiian Islands probably originate from down around here. And it's not totally conclusive, but it makes certainly sense, because Big Eye are an animal that, like yellowfin and bluefin, need warm water to spawn in, but it seems they more optimally spawn in even warmer water than yellowfin tuna, which exists down here for sure. whereas yellowfin spawns seasonally when the temperatures are adequate around the Hawaiian Islands. But why I harp on this, because now I'm on big eye tuna, is that it's really important to know where your source population comes from. If you got to manage your fishery effectively, and our Hawaiian longline fishery is mostly a big eye tuna fishery. That's what it depends on. Yellowfin, very little, and all the others utilized bycatch like mahi, wahoo. That's just small potatoes. The main thing is big eye tuna, and they catch it primarily in this north of the islands up here. Nevertheless, even though that's been that way ever since the beginning of fishery, we don't really know where those big eye come from. And so that's something we're trying to correct in working with some tagging projects to release tagged big eye tuna with satellite tags up in that northern area to try and learn more about their movement habits rather than just tag around here and hope that we get information up and down or wherever they go to. So I think, let's see. Oh yeah, this just kind of summarizes all that. People talk about our local stock. I, as a scientist, don't necessarily like the term strictly local, but I just say that primarily they have restricted movement. And the chances are of the tuna that you catch here, if you catch it here, chances are it probably had a high probability of being spawned here and a high probability of going out throughout its life in this region of the North Pacific. Big eye is not really local stock either, but probably from production south of Hawaii, so that if you want to look at more sub-regional, look at big eye management, that might be warranted, but we need more studies to really determine where that stock is born, where it travels to, and what our fishery is targeting, which group of fish is targeting. So I'll finish up with this particular fish, which I tagged on the Cross Seamount, which is just south of Hawaii, a major fishing ground south in the Hawaii zone. It was about 8 or maybe 10 pounds when I tagged it, and on that Cross Seamount. And then 7.5 years later, it was caught on the same mount at 205 pounds. So this was a really valuable animal as far as like, where have you been in the meantime? We don't believe it was on the seamount the whole time. Unfortunately, the otoliths were destroyed when the buyer cut the head off of it. And worse yet, the tissue sample they took at the same time was lost in the freezer power outage issue. So that story wasn't told, but we're still trying to fill in that gap because we know that places like Hawaii are important feeding areas for these animals. They're visited and possibly revisited over time. And we'd like to kind of close that information gap and learn. Basically, you have to know everything about the biology of an animal if you're going to manage it effectively for sustainability. This is always what we're trying to add. We're getting at, trying to get at sustainable fisheries into the future. And yeah, this is the same thing. I just point out that all this fish up here that are big eye tuna, obviously not spawning there. It's be important to find out their story because our fishery depends on them. Thank you very much. That's it.
All right, do we have any questions?
Yeah.
Hey, he said that the people that catch the fish, they get a point.
Oh, the people that catch a fish.
Yeah.
Yeah, there's a reward of some kind.
And so do they turn this tag into you, or if the fish with the tag goes to the auction, and then people that at the auction, can they see the tag too, or?
But it depends on the country and the place. In these large-scale tagging projects, we have There is a tag recovery officer in each country who kind of oversees this. There's a lot of publicity. For the big projects, we had posters printed in 20 languages and outreach, and rewards kind of scaled to the local economy. Yeah, and people, the fish is theirs to keep. We ask for the length and the date, information like that, you know, where they caught it, how they caught it. But if they turn the tag in, they get the reward. And here in Hawaii, we have a really good relationship with the auction. And the auction workers, we tell the fishermen to leave the tag in, and the workers at the auction will measure the fish. They call up, get the information, and then the reward gets back to the fishermen. Yeah.
How much are the implants? Are they like $100, $1,000?
The what?
The implants that go inside the fish so that it can be tracked?
Oh, the tags, those. The sonic tags cost about $500 each. The archival tags are closer to $1,500 to $1,800 each, and satellite tags are a few thousand. Yeah.
How long do they last?
It depends on they're all programmable, so you program them to have how many recordings per minute or whatever you do. But they can be anywhere from like 6 months to 5 years duration. Yeah. But to get a archival tag back with 5 years of complete data, that's it hasn't happened to me yet. But I have colleagues who have 3-4 year data streams, and it's an amazing amount of information.
Are the tags battery operated?
Yeah, that's the weakness of battery operated, and but they've gotten a lot better. It's pretty amazing technology. Yeah, and as batteries are getting better and smaller, and computerization is getting better, they're also getting smaller. Yes.
Even when you first 2 slides that showed the different 5 different fish.
Yeah.
Is that comparable the size? I think it was like the skipjack that was called was really tiny.
Yeah, they were scaled to each other. So bluefin tuna can get over 1,000 pounds, and skipjack are usually 3, 4, 5 pounds, up to 20 or so. Yeah. Mm-hmm. Yes.
How do you say like 100,000 fish? Is it over a period of years, or what's the period?
The period of that program, yeah, has been about 5 or 6 years. The way you do it is with these large or medium-sized Japanese pole and line boats, these sampan boats, where you have the guys pulling the tuna, and tagging stations, tagging tables, and bow and stern. I used to run a lot of these cruises, and all these boats. It's not fairly easy for these boats to do 1,000 fish a day. We did over 4,000 some days, and 2,000 is not that hard. They have live bait, they chum, like the local sampans do, and then at a 20-man crew pulling tuna onto these cradles. You have a digital voice recorder. You're just recording species, length, and then the tag number, and the tags are all lined up. It's quite a production. They're out of the water just maybe 10 seconds. Okay. Yeah. Yeah, what I was just speaking about was with these simple dart tags that are put in the back. The electronic tags that have to be put inside the body cavity. Yeah, that's a standard, like a medical sewing procedure. So use a scalpel, put it in, sew it up.
Any other questions?